Hello everyone. In the last class, we have discussed your amino acid and protein person. So today we will complete this amino acid part and protein part, and ultimately at the last in this session we will complete this. So let's come to this last person that we have left. Okay. So this is the general structure of the amino acid that we have discussed in the last class. Okay. So these are the questions that we have discussed in the last class. Protein means this is having so many amino acids. Okay, so up to this we have completed in your last class. Okay, in the last class, just wait a minute. Up to this we have completed in your last class how this protein is actually formed. How this protein is actually formed. Let me show this mechanism or general things about the protein. How this protein is formed, as you know that this is the DNA. Okay, the DNA make the DNA make the RNA, and that RNA ultimately give rise to the protein. Okay, so this is the general process. This is the general process by which what happen, the RNA that give rise to your the DNA give rise to the RNA, and RNA ultimately give rise to the protein. So if you see, the information for this protein is actually transferred from what? It is actually transferred from the DNA, but this DNA is not directly synthesizing this protein. It transcribed the RNA. First of all, what happened? It make the RNA, and that RNA will ultimately make the protein. So this whole process from the DNA to RNA and RNA to protein synthesis, this is called as what? This is called as your central dogma. This is the central dogma. What is central dogma? Central dogma, that is the unidirectional flow of information from DNA to RNA and RNA to protein synthesis. That is called as your central dogma. Okay. What is central dogma? Central dogma is the unidirectional flow of information. Okay. Let's see here. This is unidirectional because only DNA can make the RNA and RNA can make this protein. So the protein synthesizing information, that is transferred from your DNA to protein, not from the protein to DNA or protein to RNA. So that it is only occurring in one direction. That's why I'm just saying that this is a unidirectional flow of information. Which information? That is the synthesis of your protein. Okay, the synthesis of protein, that is the information. Let's see here. Let me make this clear in another slide. Okay, what happened? DNA have the nucleotide sequence. DNA have the nucleotide sequence, and that nucleotide sequence will convert the RNA, or basically we are discussing about the mRNA, and that mRNA took the help of ribosome. It took the help of ribosome, or other enzymes are all required for this process, and ultimately there will be synthesis of your protein. And the protein, that is the sequences of amino acids. The protein is the sequences of amino acids. Okay, so the sequences of amino acid is decided by the sequences of the mRNA, that is your codons. On the mRNA there is presence of your codon, and that codon, the codons are the three nucleotide sequences. Codon are the three nucleotide sequences, and each three nucleotide sequences, they decide the sequence of this amino acid, and the sequence on mRNA, that is decided by the sequence of your DNA. Okay, so sequence of mRNA, that is decided by the sequence over the DNA. Okay, so this flow of information from DNA to protein synthesis, this is called your central dogma. Okay, this is the central dogma. This whole process, that is called as what? Central dogma. Okay, So after completion of the central dogma process, there will be formation of what? There will be formation of your protein. Okay. Let's come to that slide, then we can confirm that person actually. In the last class, we have discussed the primary structure of the protein. Okay. So here, this is what? This is the DNA, and this is the sequence over the DNA. This is the nucleotide sequence over the DNA, and this is the sequences over the mRNA, and this is the sequences over your protein. So if the proteins are the amino acids are joined in a linear fashion, if they are joining in a linear fashion, then that is called as your primary structure of the protein. What is primary structure of the protein? If the proteins are joining in a linear structure, then that is called as your primary structure of the protein. Here, what happened? The proteins that is having one amino acid, then another amino acid, then another amino acid, then another amino acids. So that what happened? The amino acids are joining in a chain. The amino acids are joining in a chain. Then that kind of chain of amino acid, that is called as your primary structure of the protein. And when we see that primary structure, we can find two ends. This is one end, this is another end. Here, this amino acid having the NH2 group that is free here. The NH2 group is free here. That's why that is called as your end terminal. And this amino acid, this is the end part that is having your leucine. And here there is addition of your carboxylic group. That's why this is called as your C terminal of the primary structure of the protein. Okay. Let's see the secondary structure of the protein. Simply, the primary structure means number of amino acids that are joined in a linear fashion. They are joining in a line. They are joining in a chain. That is the primary structure. Simply, that is the very simple structure. Only amino acids are joining in a linear fashion. But when we see the secondary protein, in the secondary protein, what happened? The primary structure that undergoes folding. Okay. Suppose this is the primary structure of the protein. It undergoes foldings. Okay, it undergoes foldings, and that foldings of the primary structure of the protein, it make what? It make the secondary structure of the protein. How this secondary structure of the protein will form? When the primary structure of the protein that undergoes foldings, then it will make the secondary structure of the protein. Or sometimes we can see some of the foldings, and this folding may occur in three different ways. Okay, the protein foldings or the primary structure of the protein that undergoes the folding process in three different ways. One of the way, that is the alpha helix, another one that is the beta pleated, another one that is the triple helix. Okay, here we have to discuss three things or three kind of folding that make the secondary structure of the protein. Okay, that make the secondary structure of the protein. How this secondary structure of protein is formed? When the alpha helix is that is the secondary structure. The beta helix, the beta pleated protein, that is the secondary structure, and your triple helix, that is also the secondary structure of the protein. Okay, so let's see the first one. Let's see here, this alpha helix just looking like a spring. The alpha helix, this is just looking like a spring, that is called as your alpha helix. But this is just like a plate. Okay, this is just looking like the plate. That's why this is called as your beta pleated. And why this alpha is given, why the name beta is given, we will discuss. Okay. So, and the last one, that is the triple helix. Here there will be addition of your three polypeptide chains are added there, and they are holding together with the help of your hydrogen bonds. Okay. So let's come to that first structure or the first secondary structure, that is the alpha helix. Okay. Look, why this is called as alpha? Actually, alpha term is used because it was first discovered. It was first discovered or described. As it is first described, that's why this is alpha helix. We are using the term alpha because it was first described. Okay. So when we see the alpha helix structure, this is the primary structure of the protein, and this primary structure of the protein, it undergoes coiling, it undergoes foldings. Okay, so this is the secondary structure where it undergoes coiling, and it's just looking like your DNA. But in the DNA there is presence of double strand, but here there is, in case of your protein, there is presence of single strand. Okay, and that single strand of DNA, it undergoes this kind of coiling. Okay, and after that coiling, the structure which is formed here, this is the secondary structure of the protein. This is the secondary structure of the protein. And this helix is stable, or this helix that becomes strong due to the presence of the hydrogen bonds. There is presence of hydrogen bonds. This is your hydrogen bond. Okay, in between the helix there is presence of your hydrogen bond, so that the secondary structure of the protein that will be stable. Okay, so stability is provided by this hydrogen bond. The stability is provided by the hydrogen bond. Okay, and another things we have to remember, this helix, in this helix the protein, primary structure of the protein, it undergoes coiling. And in which direction it will coil? It will coil in right-handed direction. Okay, it will coil in the right-handed direction. So protein strand that undergoes right-handed coiling to make this alpha helix structure. Okay, and here suppose this is one piece. Let's see here. This is one complete coiling. Okay, let me choose one color. This is one coiling. This is one complete coiling. Okay, and this complete coiling is called as one pitch. This complete one coiling, that is called as your one pitch, or one torn, or one torn. And the length of this one torn, the length of this one torn or this one pitch, that is 5.4 angstrom. In case of your alpha helix, this is 5.4 angstrom. Okay, the length of this one piece, that is 5.4 angstrom. And as you know that the protein, here there will be presence of what? In the protein there will be presence of our amino acids. So suppose these are the amino acids present here, and they are making this structure. So in one helix, there will be presence of how many amino acids? In one helix there will be presence of 3.6 amino acids. In one helix there will be presence of 3.6 amino acids per piece or per turn of the amino acid. Then they are holded by hydrogen bonds. Yes, they are holded by the hydrogen bond. Okay, we will see another structure, then we can confirm that how this hydrogen bond is formed. Actually, hydrogen bond is formed between the NH group and COO group. Okay, the NH group and COO groups, the C double bond O and the NH, these are making this hydrogen bond. Okay, the hydrogen bond that is formed between the COO group and NH group of the amino acids in the protein chain, or in the alpha helix structure of the protein, there is presence of what? Amino acids. And these amino acids are held together with the help of your hydrogen bonds. Let's see how this hydrogen bond is formed. That is very simple. We have to just remember what is that structure actually. Okay, this is the alpha helix structure. Just it is looking just like this. Okay, just I'm making this like this. Okay, so let's see here. This is the protein secondary structure, or this is the alpha helix. And here what happened? Here the first amino acid have NH group. Okay, the NH group of the first amino acid is connected with the C double bond O group of the fourth amino acid. Suppose this is one amino acid, this is second amino acid, this is third amino acid, and this is fourth amino acid. Okay, so I'm just giving the numbering. So this is first amino acid, this is second amino acid, this is third amino acids, and this is fourth amino acid. Okay, the hydrogen bond will form between the NH of first amino acid, and C double bond O of next fourth amino acid. Suppose this is the first amino acid, then the hydrogen bond, this is the hydrogen bond. This hydrogen bond is formed with this C double bond O of the fourth amino acid. This is amino acid 1, this is 2, this is 3, and this is your fourth amino acid. So the hydrogen bond will form between the first amino acid and next fourth amino acids of the protein chain. Okay, so if it is first, then the first one will connect with the fourth one. If it is second one, the second one will connect with the fifth one. Or if it is third one, then it will connect with the sixth one. Okay, and so on. It will continue. Okay, so first one will connect with the fourth one. And so let's see here. This is the second one. Just wait a minute. Yes, this is the second one. This second NH group is connected with the fifth amino acid. Okay, this third NH group is connected with the sixth amino acid. Okay, then the fourth NH group, this is the fourth NH group, is connected with the C11O of the next seventh amino acid. Okay, so this is the pattern how this hydrogen bond is formed in the secondary structure of this alpha helix structure of the protein. Okay, I think you all clear about this. Okay, so this is the alpha helix structure. Simply we have to remember alpha helix means here the DNA is here the protein chain or that amino acid chain, they will just coil around their own axis. This is coiling around its own axis, and there will be formation of your hydrogen bond between these different amino acids that we have discussed. The first amino acid will connect with the fourth amino acid, the second amino acid will connect with the fifth, the third will connect with the sixth, and so on. Okay, so always the hydrogen bond will form between the NH group and the C11O group of the different amino acids. Okay. Okay. Another one, that is a beta pleated protein. Another one, that is the beta pleated. This is another kind of your secondary structure of the protein, and this is your beta pleated protein. In beta pleated protein, here there is presence, the chain is not helically coiled. Here in this structure or in this secondary structure of the protein, we can't find its helical coiling, but they are arranging in a zigzag manner. This is one chain. This is one chain of amino acid. This is one chain of amino acid. This is another one chain of amino acid. Okay. Here both of these chains that are held together with the help of hydrogen bond. This is hydrogen bond. This is hydrogen bond. Okay. One thing we have to remember, always the hydrogen bond will form between the NH and C11O. Here there will be presence of your C. This is C. This is C, C. Okay, and so on. This is carbon. Okay, so this is one protein chain. This is another protein chain. In between this protein chain, there will be formation of your hydrogen bond. Okay, so they are just looking like this manner. They are just arranged in this manner. Why that is called as pleated? Beta pleated means they are just looking like a plate. Just imagine. These are just looking like a plate. Okay, actually this is the 2D structure. The beta pleated is not able to form your 3D structure. It is forming 2D structure. The beta pleated, it is forming 2D structure. And why this is beta? Because it is discovered after your alpha helix structure of the protein. Okay, here there will be presence of your two chain, and in between this chain of your amino acid, there will be formation of your hydrogen bond, so that it gives the strength for their joining. Okay, and if you see the length of one peach, this is one peach. Let's see here. Let me make this another color. This is one peach. Okay, this is one peach, and the length of one peach. Okay, this is one peach. Actually, this is one peach. It start here and it end here. This is one piece having the 7 angstrom. The one piece of the amino acid, the beta pleated structure of the protein, that is having 7 angstrom. Okay. The last one, last model or the last structure of the protein, that is the triple helix structure of the protein. This structure was given by Ramachandran. This structure was given by the scientist Ramachandran, and this is a rare form of protein. The triple helix structure of the protein, that is a rare form of protein. Okay, and it is having 3 polypeptide chain. This is having 3 polypeptide chain. This is first polypeptide chain. This is second polypeptide chain, and this is third polypeptide chain. There is presence of how many polypeptide chain? 3 polypeptide chain. Okay, and these polypeptide chains are held together with the help of your hydrogen bond. These are the hydrogen bonds. Okay, this is the hydrogen bond. Okay, so here in this structure also they are held together with the help of your hydrogen bonds. Okay, sorry for the disturbance. So if there is presence of three polypeptide chain, and these 3 polypeptide chains are held together with the help of your hydrogen bond, then that kind of structure of the protein, that is called as your triple helix structure of the protein. This is called as your triple helix structure of the protein. And one of the example where this triple helix structure is found, that is the collagen fiber. This is the one of the protein that is 1/3. Okay, 1/3 of the protein of human. 1/3 of protein of human is collagen fiber. It is the collagen fiber, and this collagen fiber, it gives strength to the different parts. It gives strength to different parts of the body, as well as this collagen fiber, it gives elasticity to the skin. It gives also elasticity to the skin. Okay, so in case of your collagen fiber, this is one of the most abundant protein that is found in case of the human body, that is the collagen fiber. And that collagen fiber having how many polypeptide chains? There will be presence of 3 polypeptide chains, and these 3 polypeptide chains are held together with the help of what? With the help of your hydrogen bonds. Okay, so this is the triple helix structure of the protein. That is very simple. The first one, that is the alpha helix. This is the alpha helix. In the alpha helix, just it is coiled around its own axis. Okay, and the 1 feet of the alpha helix structure of the protein, that is 5.4 angstrom. We have to remember that. And in that 5.4 angstrom of the protein, 5.4 angstrom of that alpha helix, there is presence of how many amino acids? There is presence of 3.6 amino acids. Okay, and hydrogen bond is formed between the C double bond O and NH group of the different amino acids. And in that part, we have discussed that the first amino acid will connected with the 4th amino acid by the presence of your, or by the help of your hydrogen bonds. Okay, and here there will be NH, and here there will be C double bond O. Okay, that we have discussed. And this is the beta pleated. Here 2 chains are added together with the help of your hydrogen bonds, or another chain may added here. Okay, just I am discussing about the 2 chains, but another chain may added here to make this beta plated structure also. Okay, suppose another chain is added here, and in between them there will be formation of your hydrogen bond. This is the beta plated structure also. Okay, so it is not fixed that always there will be addition of only 1 chain, only 2 chain. There will be chances of your joining of more than 2 chain also in case of your beta plated structure of the protein. Okay, and this is the triple helix, where there will be presence of your 3 polypeptide chain, and these 3 polypeptide chains are held together with the help of your hydrogen bond. Then the tertiary structure of the protein, we have to discuss the next folding. If the protein that undergoes next level of folding, means the secondary structure of the protein further folds to form the tertiary structure. In the last one, up to your last one, only hydrogen bond is formed in the helix. Okay, whether that may be your alpha helix, or that may be your beta plated, or that may be your collagen fiber, or that is the triple helix. But in case of your tertiary structure, what will happen? There will be further foldings. The secondary structure of the protein, it may undergo further foldings to form the tertiary structure. Okay, and in that folding, what will involve here? in that folding, there may be, first of all, there will be formation of your hydrogen bonds. Yes, the hydrogen bond is formed between the C double bond O and NH group. Okay, and another one, if there is presence of your cysteine, if there is presence of cysteine, cysteine is a sulfur containing amino acid. Cysteine is a what? Cysteine, it is a sulfur containing amino acid. Cysteine is a sulfur containing amino acid. Okay, so what will happen if there is presence of your sulfur, and if there is presence of one cysteine here and another cysteine is present here, then there will be attraction between this sulfur of the cysteine 1 and cysteine 2, and between them there will be formation of a bond that is called as your disulfide bond. Okay, first of all, there is formation of what? First of all, there is formation of your hydrogen bond between the C double bond O and NH group of the amino acids. Okay, this is one amino acid, this is another amino acid, and they are held together with the help of your hydrogen bond. Okay, but when, if there is presence of your cysteine in that amino acid chain, okay, this is one cysteine, this is another cysteine, then in between the cysteine there will be formation of your disulfide bond. This is the disulfide bond formed between your two cysteine in the amino acid chain. Okay, or there will be formation of your ionic bond. As you know that if we put the amino acid in the solution, then this will convert it into your COO minus, or it may convert it into NH3 plus. Okay, so in between this COO minus and NH3 plus of your, this is one amino acid, suppose this is another amino acid having this COO minus and this is having NH3 plus. In between them there will be formation of your ionic bond also. Okay, or if the R group is hydrophobic, hydrophobic means it is not mixed with the water, it is not soluble in water. If the R group is not soluble in water, or that is hydrophobic substance, then another R group is there and that is also hydrophobic substance, then what will happen? In between the hydrophobic substances or hydrophobic R group, there will be formation of a attraction, and this attraction is called as your hydrophobic attraction. This attraction, that is called as what? Hydrophobic attraction. Okay, so if this is the protein, here also there will be formation of other bonds except your hydrogen bond. There will be formation of other bonds, so that it will coil more and more, it will coil more and more. So in the first of all, this is the linear, then it undergoes the first level coiling or folding, and it make what? It make the alpha helix, and when there will be formation of your other bond between them, then it will further coil and it will look just like this. Okay, so further attraction between the different amino acids will occur, and attraction that may be due to the hydrogen bond, or that may be due to the disulfide bond, or that may be due to the ionic bonds, or that may be due to the hydrophobic attraction between the R group. Okay, so due to that, the protein is go on further, it is undergoing the process of your folding and refolding, and ultimately it form the another structure, another level of coiling will there, and that structure of the protein, that is called a tertiary protein. Okay, this is the tertiary protein. And the last one, that is the quaternary structure of the protein. This is the most complex form of the protein, the most complex form. Okay, this is linear, this is the helix, then this is your tertiary, and the last one, that is more coiled structure, that is called as your quaternary structure of the protein. Okay, this is the quaternary, and this is the most complex form, and this is a macromolecule. Okay, and it only formed by oligomeric protein. What is oligomeric protein? Quaternary structure. This is one tertiary structure. Okay, and what will happen? Let's see here. Quaternary structure. In the quaternary structure, there will be involvement of oligomeric protein. Oligomeric protein means what? If there is involvement of more than one polypeptide chain. This is one more than. This is one polypeptide chain. This is another polypeptide chain. This is another polypeptide chain. This is another polypeptide chain. Suppose there is addition of your 4 polypeptide chains and that are connected with each other, so this is the quaternary structure of the protein. For example, in case of your hemoglobin, okay, oligomeric means what? Oligomeric means, just let me write here, oligomeric means the protein having more than one polypeptide chain, having more than one polypeptide chain. Okay, that is called as your oligomeric. This is one polypeptide chain. This is another. This is another. This is another. In case of your hemoglobin, what happened? There is presence of 4 polypeptide chain. Okay, hemoglobin, as you know, that is a part of our blood. Okay, in the blood there is presence of your hemoglobin, and this hemoglobin, that is the oxygen transporter. This is the oxygen transporter in the blood. It carry the oxygen to the different tissues. Okay, so the hemoglobin, that is the oxygen transporter, that is having 4 polypeptide chains, and there will be presence of 2 alpha chain and 2 beta chain. Okay, suppose this is one alpha, this is another alpha, this is beta, and this is another beta. Okay, and all these proteins that are held together here, all this protein, and these 4 proteins are connected with the iron. Actually, we will discuss in the next diagram. So each chain undergoes folding and attract towards each other. This alpha chain, this is simply the linear structure of the alpha. It undergoes folding, and again it undergoes next level folding, and ultimately it makes this structure. And this one, this is the alpha. Another alpha will form, another beta will form, another beta will form, and they are attracting towards each other. They are attracting towards each other, and they are making a 4 polypeptide structure, that is called as that is the hemoglobin. And here each structure, each unit of this 4 polypeptide chain, that are called as your protomere. Okay, the single unit of the hemoglobin, that is called as your protomere. This is single unit, this is another unit, this is another unit, this is another unit. Okay, so these are the protomere. Okay, let's see here. This is beta globin chain, this is another beta globin, this is beta, this is alpha, and this is also the alpha. Okay, all these are held together and that are connected with the iron, that are connected with the iron, or Fe2 floss, that are connected with the iron. Okay, so here this is the quaternary structure of the protein. This is what? This is the quaternary structure of the protein. And in the quaternary structure, there will be presence of your the oligomeric protein. Means this is one polypeptide chain, this is another, this is another, this is another. Okay, so that this is your quaternary structure of the protein. But here all these protein that are compactly arranged, or that are coiled in a different level of coiling, that leads to formation of this structure actually. Okay, then the last portion of the protein, that is your function of the protein. Protein that may helps in the transport. The protein may helps in the transport, so that are called as your transport protein. Okay, and as you know that the molecules are transporting from one area to another area in the body, that is by the help of your protein, that is called as your transport protein. And one of the example, that is your hemoglobin, it transport the oxygen and carbon dioxide. Hemoglobin will pick up the oxygen, and that oxygen will transport to the different tissues. The hemoglobin will transport to the different tissues of the body, and that hemoglobin transport what? Transport the oxygen to the different tissue of the body. That is your transport molecule, that act as transport molecule in case of your human body. And another one, that is the glucose transporter. Before that, let me clear something here. Suppose this is the plasma membrane. This is the plasma membrane. And as you know that in the plasma membrane, there is presence of what? There is presence of our phospholipid bilayer, and protein is also there. Yes, and different proteins that are present above here, or the lower side also there is presence of your protein. And as well as here also there is presence of some proteins, and some protein that also penetrate in this both phospholipid bilayer. This protein, that is called as a transmembrane protein. This is called as what? Transmembrane protein. Okay, what is the function of this transmembrane protein? This transmembrane protein, it allow, suppose here there is presence of some molecules outside the plasma membrane, then it will allow the molecules to pass through these, okay, from the outer side to the inner side, that is your transmembrane protein, or this is also called as your carrier protein, or it may call as your carrier protein. This carrier protein, it carry the molecule from the outer side, or it may carry the molecule from the inner side to the outer side, or from the outer side to the inner side of the plasma membrane. So here also the protein that act as a carrier, it carry the molecules from the outer side of the plasma membrane, and it will just move this towards the lower inner side of the plasma membrane. So that is called a transporter protein, or we may call them as carrier protein. Okay, and sometimes it acts as glucose transporter. What is glucose transporter? Let me clear you. Glucose transporter. Glucose transporter, this is also a protein. From this name we can clear that if this is the cell, and outside the cell, suppose there is presence of our glucose, then that glucose has to enter inside the cell. That glucose has to enter inside the cell. But this glucose will not easily enter inside the cell without any help. Okay, it will take the help of some protein, and some proteins which are present in the plasma membrane. Some proteins that are present in the plasma membrane, and these proteins that are glucose transporter. This protein that are the glucose transporter. Okay, suppose these are the glucose transporter. These are glucose transporter protein. We may say them as glucose transporter protein, and that glucose transporter protein that will allow the movement of your glucose from the outer side. Suppose here there is presence of your glucose. The glucose can only pass. The glucose can only pass through this membrane, through this what? Through this transporter protein, that is the glucose transporter. It cannot pass through this part. If there is no glucose transporter, then the glucose will not able to enter inside the cell. Okay, if there is no glucose transporter, then the glucose outside the cell will not able to enter inside the cell. That is the glucose transporter. And all we know that always we require the glucose for the generation of energy. For the generation of energy, there will be requirement of your glucose, and that glucose has to be enter inside this cell through the transporter protein, that is the glucose transporter protein. Okay, so this is the function of the protein in case of your cell. Another function, that is transferrin, one of the protein that is called as transferrin. In that transferrin protein, it transports the iron in the blood. The iron plays a vital role in the blood, and that iron is actually transported through this protein that is called as a transferrin protein. Okay, so these are the different functions of the protein. Okay, and here I'm making this structure, and this is the structure of different amino acids. Okay, as you know that in the nature there is presence of several thousands of amino acids, but the protein are only made up of the 20 amino acids, and these 20 amino acids are glycine. Okay, this is one. Then the alanine, then serine. This is the structure. Then threonine, then your cysteine, then your valine, then this is your leucine, this is your isoleucine, this is your methionine, this is your what? This is the proline, this is the phenylalanine, this is your tyrosine, this is your tryptophan, this is your aspartic acid, this is your glutamic acid, this is your asparagine, this is your glutamine, this is your histidine, this is your lysine, and this is last one, that is the arginine. These are the 20 different kind of amino acids. These are 20 different kinds of amino acids that is that are making the protein. Okay. Okay. So one of the example, let's see here. One of the example, this is the structure of insulin. This is the primary structure of insulin. primary structure of the insulin. Okay, actually the insulin that is having two polypeptide chain. The insulin that is having your two polypeptide chain. This is polypeptide chain 1. This is polypeptide chain 1. Okay, and this is polypeptide chain B. Oh, sorry. Anyway, here we may write this. This is chain A and this is chain B. Okay, so this is polypeptide chain A. This is polypeptide chain B. And in the chain A, there is presence of how many amino acids? There is presence of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Here there is presence of how many amino acids? 21. And here in the B chain, there is presence of your 30 amino acids. Okay, here there is presence of your 30 amino acids in the B chain and 21 amino acids in the A chain. And all these are interconnected with the help of your peptide bonds. These are interconnected with your peptide bond. That's why that is your polypeptide chain. Okay, and as you know that the cysteine, this is the cysteine. The cysteine, that is what? That is the sulfur containing amino acid. This is the sulfur containing amino acid, and it is having sulfur. And here another cysteine is present. In between the cysteine and this cysteine, there will be formation of your disulfide bond. So that the structure that may be just looking like this. The structure will be like this. Here, this is one cysteine is present. Here, this is suppose cysteine is present. Then here there will be formation of your disulfide bond. In the secondary structure, we can see this. Okay, in this secondary structure, we can see this. Okay, because there will be attraction or there will be formation of this bond. Due to that, it will fold. Later on, it will undergoes folding. Then in between these two chain, here in this chain there is presence of cysteine, and here also there is presence of cysteine. So in between them there is also formation of your disulfide bond. This is the disulfide bond. Okay, and here another cysteine is present and another cysteine is present here also. So here also there will be formation of your disulfide bond. So A chain and B chain that are held together with the help of which bond? That is your disulfide bond, and this is the primary structure of your insulin protein. Insulin, that is the glucose transporter protein, or it is just transport the glucose, or it gives the information to the different cells about the presence of the glucose in the blood. Okay, so the glucose transporter will active, and that glucose transporter will allow the movement of glucose inside the cell. That is the insulin, that is the activator. That is the activator actually. That is not a transporter. That is an activator. It activates the cell to make its glucose transporter that should open. The glucose transporter protein will open so that the glucose from the outer side or in the blood that will enter inside the cells, so that the cell will get the glucose and it will make the energy. Otherwise, if in our body the insulin level is become lower, then the glucose level in the blood will increase. If the glucose level in the blood will increase, that is called as your diabetes. That is called as your hyperdiabetes. That we know that. But if the insulin level is good, then what will happen? The insulin just allow the glucose molecule to enter inside the cell. Okay. Okay. Another things we have to just discuss here, that is your isoelectric point of the amino acid. The last person, and after completion of this, I will complete this session. Okay. So here we have to discuss the isoelectric point. Okay. Let's see the isoelectric point of the amino acid. What is the meaning of isoelectric point of amino acid? As you know that the amino acids, if we put the amino acid in a solution, okay, in a solution, the amino acids that are exist in three different forms, they will available in three different forms, okay. One form, that is the acidic form. The second one, that is the basic form, and the third one, that is the neutral form. Okay, so let's see here. Suppose this is the one amino acid, it is having the R, then there will be presence of your NH3 plus COOH. Okay, and this is the hydrogen. Suppose this one is the one form. Another form, the amino acid may exist in this form also, COO minus NH3 plus, this is R group, this is hydrogen. Or it may exist in this form also, C double bond O minus, then NH2, this is your R, this is the H. Okay, and all these are interconvertible. Okay, so if we just took them in the water, it may exist in this form. Okay, here if it is existing in this form, then this part will donate the hydrogen because it is NH3 plus. This part can donate the hydrogen, and this part also can donate the hydrogen. Means this is the acidic, already the COOH, that is the acidic, and the NH3 plus, it can also donate the hydrogen, and the substance that donate the hydrogen, that is called as your acidic. So that this form of your amino acid, that is the acidic form. This is the acidic form, and here this COO minus, this is become electron donor. This will become what? Electron donor. And if the substance that can donate the electron, then that will be what? That will be our your basic. And this is already basic. This is the basic, and this will become basic because it can donate the electron in presence of water. So this form of your amino acid, that is called as your basic form. Okay, but if it is existing in this condition, that means it is NH3 plus. Okay, it is NH3 plus, and this is COO minus. Okay, that is having both positive and negative charge. This is having both positive and negative charge. Then this form is called as your neutral. This is called as your neutral form. The net charge of this amino acid, that will become 0. The net charge will become 0 because there is presence of both same amount of your positive charge and same amount of your negative charge, so that the plus minus will cancel, and due to that the net charge of the amino acid will become what? That will be your 0. And this form, that are the ion, if any substance it is forming both positive and negative charge, then that kind of ion is called as your Zwitter ion. That is called as what? Zwitter ion. In the last class also I have discussed that part. So here, if there is presence of same amount of NH3 plus and COO minus, here both positive and negative charge is there. That's why that is called as your neutral form. And here the net charge of this amino acid will become what? That will become 0. Okay. So when this acidic form or this neutral form or this basic form, that will dominate. Okay, when the basic form will dominate, or when this basic acidic form will dominate, or when this neutral form will dominate. Okay, when they will dominate, because let's see here, if we put this amino acid in an acidic medium, if we put the amino acid in an acidic medium, in the acidic medium, there will be what happened? The acidic form will exist more. If we put this in the acidic medium, the acidic form of the amino acid will dominate. If we put this amino acid in a basic medium, then this form will dominate. Okay, suppose we put the amino acid in pH 0. Suppose in pH 0, if we put the amino acid in pH 0, that is the acidic medium. The pH 0, that is the acidic medium. Here, in the acidic medium, this is already acidic, and this NH3 plus will also release the hydrogen ion, so that there will be formation of more amount of your hydrogen ion that hydrogen ion can remove or that can donate by this COOH as well as your NH3 plus. So in that acidic medium, the acidic form will dominate. in the basic medium, the basic form will dominate. But when we put this in the basic medium, means let's put them in the pH 10. In pH 10, it will dominate by your basic form. In pH 0, it will dominate by your acidic medium. But when we put this molecule or this amino acid in pH 7, suppose in pH 7, that is the neutral pH. Okay, in pH 7, that will there will be the amount of your positive and negative charge will become what? That will become same. Okay, or there will be formation of your deuter ion. Okay, so it is not fixed. I am not giving the standard pH in which pH there will be more acidic form, or in which pH there will be more basic form, or in which pH there will be more deuter ion form of the amino acid. That is not the standard. This is not 0 or not 10 or not 7. For different amino acid, this value is different. Okay, anyway, I want to say that what is isoelectric point? The isoelectric point, it is the pH at which there will be formation of your more deuter ion from the amino acid. What will be how many amount of your, or the maximum amount of your deuter ion will form at which pH? That is called as your isoelectric point. What is isoelectric point? It is the pH at which the positive charge on the amino acid is equal to the negative charge. Okay, the positive charge and negative charge that are same at which pH? That is called as your isoelectric point. Iso means same. Iso means what? Iso means same. What is isoelectric point? The point at which the amount of positive charge and negative charge that becomes same, or at this pH, the net charge will become 0. The net charge will be 0. Okay, or at which pH the net charge of the amino acid becomes 0, that is called as your isoelectric point. Okay, so hence what will happen if we put the this form of your amino acid in the any medium, then it will not move towards the cathode or anode. They will not move towards the cathode or anode. Okay, they will not move towards the cathode or anode because the net charge is 0. Okay, if it is a negative charge, then it will move towards the cathode. If it is positive charge, then it has to move towards the anode. Okay, so this is the isoelectric point. Okay, simply isoelectric point. In the biological science, I'm not discussing more thing about this isoelectric point. If we go to the chemistry, in that chemistry you can see the isoelectric point of different molecules. I am just only discussing about the isoelectric point of the amino acid, but there are several molecules which have the isoelectric point. Every molecule has the isoelectric point. That means the isoelectric point is the point at which that molecule can have the both same amount of positive and negative charge in it. That is called your isoelectric point. Okay, but if we go more details in case of your chemistry, then you can see some value, that is the pKa value of the molecules. Okay, and first we have to know the Ka value, then you can go for the pKa value, that is the minus logarithm 10 base of your Ka, that is the pKa value, and that Ka value, that is the dissociation constant of the any acidic substances, that is called your Ka value. You have to know the Ka value, then you have to know the pKa value, then that pKa value will give rise to give rise to your isoelectric point. Okay, so I'm not discussing about that more thing about your chemistry. Just for your biological student, for my biological student, I am discussing about these few things about your isoelectric point. So as a biological student, you may remember simply that it is the pH at which the positive and negative charge on the amino acid that is equal or that is same, that is called as your isoelectric point. That is called as your isoelectric point. So in the next class, we will discuss the next portion of your biomolecule, that is the lipid. Okay, in the next class, we'll discuss the